I'm not sure if I'm pronouncing right. Aude from INRA-LEPSE, France. And she's going to be talking about breeding grapevine for an efficient use of water by lowering nighttime transpiration. I want to make one point here. The abstract that is given in the abstract book is not correct. So it is fully correct in the online website, okay? The PDF on the website will have the right abstract. Go ahead, Aude.
Thank you. So, good morning, everyone, and thank you very much to the organizers for giving me the opportunity to present you part of my PhD work that I led on the grapevine species and where I explored whether reduced nighttime transpiration could be a relevant target to breed for high water use efficiency. So it's been clearly described today that maximizing water use efficiency is an important breeding target in the context of drought. But what is even more important is to maintain yield at the same time. And the main problem here that has been described by Professor Farquhar yesterday relies on the tight coupling that exists in the daytime between photosynthesis and transpiration rate through the stomata. So that closing the stomata in order to decrease the water loss will necessarily impact the carbon gain and vice versa. But by contrast, during the night, the water loss are not coupled to any carbon gain, precisely because in the absence of light, photosynthesis is inactive. So the hypothesis we explored here was whether reducing nighttime transpiration could be a relevant target to improve water use efficiency. So historically, nighttime transpiration has been largely neglected, but many recent studies led on many species have shown that plants do lose water during the night, and that these losses can account for up to 30% of the daytime rates. And they've always shown that stomata are not totally closed during the night. But apart from that, we don't know much about nighttime transpiration. First, we don't know about its genetic architecture in any cultivated species. We don't know either which are its physiological bases, and importantly, we don't know to which extent its variation could impact the whole plant water use efficiency. So these are the three questions we wanted to explore here, and we worked on grapevine using a mapping population from a cross between the two cultivars Syrah and Grenache that are well known for their contrasting water use strategies. So the potted plants were studied in a 2-year experiment by means of a high-throughput phenotyping platform in Montpellier, where they were submitted to watering scenarios. So a well-watered one and a moderate water deficit. And when both treatments were stabilized, the plants were taken off the platform in greenhouse and placed into a controlled environment chamber with stable and repeatable conditions during a 24-hour cycle, so night and day. And therein, by weighting the bagged pots, we access the water loss during the night and during the day, and we then calculated nighttime and daytime transpiration rates that were normalized by the leaf area of each plant. So we found a wide range of genetic variability for nighttime transpiration under both watering scenarios. And on average, the nighttime losses accounted for a 9–10% of the daytime ones. This ratio was also under tight effect of the genotype, and it could reach up to 30% under water deficit conditions for some genotypes. And interestingly, this ratio, the ranking of the genotypes for this ratio was well conserved across the watering scenarios, meaning that those genotypes that lost a high proportion of their water during the night as compared to the day and the well-watered conditions also did so under water deficit. We then wanted to validate to assess the validity of these results in more natural climate conditions. So we repeated the experiments on a subset of genotypes outdoors. And again, we found that the ranking of the genotypes was well conserved for the ratio of night over daytime losses between the outdoors experiment and the chamber one. We then undertook a Qtl detection for Nye Transpiration, and we detected several Qtls that were stable between the years of experiment. So we just showed that nighttime transpiration is under tight genetic control. The next question was then to explore which could be the physiological basis for such genetic variation. And we first explored a possible role for the stomata. So we switched to experiments on detached leaves that were perfusing in solution, and we first measured nighttime transpiration in a control solution for which the ranking of the genotype was consistent with the ranking observed for the whole plants. And the leaves were then transferred to an ABA solution, which strongly and significantly decreased nighttime transpiration, thus showing that the stomata were not totally closed during the night in the control solution. We also found a highly significant effect of the genotype for the stomatal contribution, the residual stomatal opening during the night, and that on average, this residual opening of the stomata accounted for 70% of nighttime transpiration. So we then wanted to explore a major role for the stomata. We then wanted to explore the missing part of this variability, and we tested a possible role of the water loss through the cuticle. And we took advantage of the hypostomatic feature of grapevine, which means that the stomata are only present on the abaxial face. So by waterproofing that face, we had access to the water loss through the cuticle of the adaxial face only. As expected, the treatment strongly reduced night transpiration, but we could still record losses, and we identified a highly significant effect of the genotype on those cuticular losses. And we also found that the total cuticular conductance might account for 30% of the total night transpiration, which means a secondary role for the cuticle, but not that negligible. So having shown that night transpiration is under tight genetic control with a strong role of the stomata, next question was to assess if there is any possibility to find genotypes with low night transpiration, but maintaining high transpiration in the daytime in order to maintain carbon gain. If we look at the QTLs of both transpiration rates, we can first see one colocalization between a QTL of night and daytime transpiration, consistent with a significant correlation between both traits, which at first sight is not exactly good news, because that suggests that if we choose genotypes with low night transpiration, they may also have low daytime transpiration and then low carbon gain in the daytime. But actually, the good news is that most of the QTL of night transpiration were independent from those of daytime transpiration, and that the correlation, although significant, was not tight at all. So that, yes, there is a leeway by acting on those QTLs to find, for a given level of daytime processes, genotype with low night transpiration. We then measured the whole water use efficiency, taking advantage of the platform to measure the growth of the plants during a whole period of 15 days and the cumulative water loss in the same period to calculate the whole water use efficiency. And we detected QTLs. What we can see is that, as expected, when at the QTL, when we have a colocalization between night and daytime transpiration, we found no QTL of water use efficiency, which means that the variation in night transpiration is not beneficial to water use efficiency when it's associated with daytime transpiration. But most importantly, we found many colocalizations between water use efficiency and night transpiration alone, which evidence that both processes share their genetic control. And if we focus on these common QTLs, and if we look at the allelic effects, we see that the alleles that confer the lowest night transpiration also confer the highest water use efficiency. And this pattern was the same for the 4 colocalizations. And even more interesting among these 4 QTLs is this one, in which we also found a QTL of growth. And the interesting thing is that we found a negative correlation between nighttime transpiration and growth, which means that by selecting the QTLs of the alleles, conferred low nighttime transpiration, you improve growth. So it's a double way acting on this QTL to improve water use efficiency. So these QTLs appear as interesting targets in the view of a marker-assisted selection program. So we have seen that nighttime transpiration is under tight genetic control and that it's partly independent from daytime transpiration. And we have seen that grapevine could possibly be bred for higher water use efficiency by selecting those genotypes with lower Nye transpiration. And that the physiological origin of the genetic variation in Nye transpiration mostly relates to the residual stomatal opening and secondarily to the cuticle. So this is it. Thank you very much for your attention, and thank you to all the people that contributed to that work.